The overall goal of this assay is to assess the anticoagulant properties of endothelial cells using whole non-anticoagulated blood.
This method can be used to answer research questions which are related to cardiovascular research, transplantation, and surgery-related research. Essentially, it can be used for all questions which have to do with innate immunity and the activation of endothelial cells.
The main advantage of this technique is that whole blood without anticoagulants can be used, as anticoagulation is guaranteed by endothelial cells.
To begin the protocol, mix 7 milliliters of microcarrier beads with 42 milliliters of collagen solution in a 50 milliliter tube and incubate the beads for 1 hour at room temperature. After incubation, wash the beads with 25 milliliters of phosphate buffered saline, or PBS, and rinse the beads by pipetting up and down. Wait until the beads settle at the bottom of the tube before discarding the supernatant and repeating. Next, wash the beads 1 time with 25 milliliters of Delbecco's modified Eagle medium, DMEM. Then, cover the beads with 10 milliliters of medium 199 and allow them to equilibrate for 10 minutes before further use. Remove the cell culture media from the T175 flask containing PAEC and add 5 milliliters of PBS. Then, remove PBS from the T175 flask and replace it with 5 milliliters of trypsin with 0.05% EDTA.